Welcome to Cladistics, Classification and Common Ancestry. What we're going to be looking at is how and why certain organisms are classified together. We're going to be looking at how you read cladograms in evolutionary trees, as well as a little bit about the history of cladistics. Now, cladistics is the modern method for making evolutionary trees. The process uses several different types of scientific evidence, such as living species, the fossil record, comparative anatomy, genetic data, and many other factors as well. Let's take a look at an example cladogram. On this cladogram, we have lemurs, monkeys, chimps, and humans. Now, all of these organisms belong on this cladogram for one reason. They all have an opposable thumb. This means that you can take your thumb and touch it to the tips of each of your other fingers. This is a great way for grasping and holding on to things, which is very important for all of these organisms. Now, the more derived characteristics that two species have in common, the more closely related they are. As I said, all of these creatures have an opposable thumb, which means that they have something in common. So there's at least one derived characteristic that they all share. However, you'll notice that each of these organisms eventually creates its own branch off of this cladogram. Where these branches come off are called nodes. A node represents the most recent common ancestor of a clade. What this means is that at some point millions of years ago, lemurs, monkeys, chimps, and humans shared a common ancestor that had an opposable thumb. However, environmental stresses and limiting factors applied to that common ancestor over millions of years will lead to new adaptations in response to those changes in the environment. As those genetic changes and adaptations occur, new derived characteristics are going to pop up that are going to start separating the different clades within this cladogram based on those new adaptations. In this case, we look at number 2, the development of a prehensile tail. Now, lemurs do not have a prehensile tail. Monkeys, however, do. A prehensile tail is a tail that almost acts like a fifth hand. It allows them to actually grasp and hold on to things so that they don't fall out of trees or they don't lose their balance so easily. Lemurs, again, do not have a prehensile tail, which is why at this point in the cladogram, they shoot off on their own branch. Because after the opposable thumb derived characteristic, they do not have any of the other derived characteristics that are on this cladogram, making them their own unique species. Each node represents the development of a new derived characteristic, and therefore the development of a new species, and on the whole, the development of a new clade. A clade is a group of organisms that are believed to share a common ancestor. All the organisms on this cladogram had a common ancestor with an opposable thumb, and because of that are part of the green clade, which we can see starts at the lemur and then includes everything beyond the lemur. At the top of the cladogram is a clade in blue. Only chimps and humans are in this clade because they share all of the derived characteristics leading up to it, meaning they have an opposable thumb, the development of a prehensile tail, and then the loss of a prehensile tail. As you can see, chimps and humans don't actually have tails. However, humans and chimps and all of the great apes do have the genetics for a tail within their genome. As a matter of fact, some humans are still born with tails. It's highly uncommon, but it does still happen. Now, here's the big question. How do we know this cladogram is correct? How do we know the development of a tail has come before the loss of a tail? Well, we have the fossil record and, more importantly, genetics to thank for that. By comparing the human genetic code to that of other species, we can actually count the number of amino acid differences or mutations between the human genome and the genome of other organisms and see how many mutations have occurred over the course of millions of years. Comparing that with the first appearance of those organisms within the fossil record, we can actually estimate how long it takes mutations to occur over time. And then we can use that as well as other information like mass extinctions, volcanic eruptions, meteorite impacts, and those sorts of things to actually determine just how old or how young a species is. The older a species is, the further down on a cladogram it will be. This means that its traits and derived characteristics are older than the ones that appear later. If we look at the number of mutations that a shark has in comparison with our DNA, we can see that there are far more mutations between us and sharks than there are, say, between us and mice or even horses. This means that sharks are far older than we are. You can see from the chart that the fossil record backs up this finding. Carbon dating shows that sharks first appeared in the fossil record over 450 million years ago. This means that sharks are even older than dinosaurs because dinosaurs didn't show up until around 68 million years ago. So we know that the cladogram above is correct because modern science techniques like genetics and carbon dating allow us to evaluate it in multiple ways. However, this wasn't always the case. As early as the 1700s, cladistics, taxonomy, and other classification practices didn't exist. In the early days of scientific development, there was a lot of competition. This is because new discoveries would lead to recognition, fame, and wealth, very much like it still does today. Now, in the early 1700s, this led to a lot of confusion, especially when it came to identifying new and existing species. The issue? Common names. As you can see from this chart, a single species can have several common names. It all depends on who you talk to and where you are. Consider the mountain lion. The mountain lion in northern and western parts of the United States is often referred to as a mountain lion. However, in more southeastern states, it's referred to as a puma or even a cougar. This led to scientists believing that there were far more species than there actually were. Enter Carlos Linnaeus, aka the father of modern taxonomy. To help fix this identification issue, he developed a new naming system based on Latin, the scientific language of the day. This system is called binomial nomenclature. It gave organisms two names that were based on their species and described what they look like. Here you can see a red fox. That's its common name. Under Linnaeus' system, it now has a scientific name, Vulpes vulpes. This is the name it will have in scientific papers and ledgers to cut down on the confusion caused by common names. Vulpes vulpes literally means fox, fox. Vulpes was the term used to indicate a fox in Latin. As the red fox was, and still is, the most commonly recognized fox, the term became its descriptor as well, Vulpes vulpes. Another good example is the barn owl. There are several different species of barn owl, and all have different markings. Some of those markings are only slightly different from species to species, so having that second descriptor name is incredibly important. In this example, Tyto refers to the barn owl family, but alba means white. So if you were a scientist out looking for new species and you came across a barn owl that was predominantly white, especially on the face and chest, you would know that you're looking at Tyto alba, not a new species. Linnaeus didn't just give us binomial nomenclature. He also developed the 7-level taxonomy system, a system that would eventually lead to modern cladistics. Now, Linnaeus' classification system had 7 levels that were based on similar characteristics. For example, the kingdom Animalia. All of the organisms that you see in that kingdom are animals, meaning they're not plants and they're not bacteria. Each level within this taxonomy system is included in the level above it. Consider the order Carnivora. All of the organisms that you see in that order are carnivores, meaning they eat meat, but they're also mammals, chordates, and animals. As you move down the chart from kingdom to species, the descriptions become more and more specific, to the point that fewer and fewer organisms can fit into each category. This means that by the time you get to the bottom, to species, there should be only one organism left, that particular species that fits with all of the descriptors that come above. So here we have Canis lupus, which is the gray wolf. The gray wolf is a canine, but it's also a carnivore and a mammal and a chordate and an animal. And therefore, by the time you get to the very bottom, the only thing that's left in the species bracket is the species that fits all of those descriptors. Now, there is a problem with this system. It's based almost entirely on visual characteristics and comparative anatomy. This is because genetics didn't exist in Linnaeus's time. As a matter of fact, the entire area of genetics within science is only a few decades old. What this means is that many organisms were classified together that shouldn't have been. Many of those organisms we now know actually belong to other groups or their own groups entirely. And this was incredibly common. So common that we're still fixing many cladograms today based on new genetic data as well as fossil records. So if we look at this example, family Canidae, all of these organisms under Linnaeus's rules were categorized together. However, one of these animals is actually more closely related to cats and the mongoose than they are to canines. That animal? The hyena. Hyenas actually have more genetically in common with mongooses and cats than they do with canines. But now, thanks to modern science and scientific technology, such as carbon dating and genetic testing, we can now fix many of these issues that we've been seeing within cladistics. Thanks for watching, and I'll see you next time.